So now it gives me great pleasure to introduce Esme Brinsden from the University of Southampton. The way we find our speakers, we try to keep alert as to what's going on, and I read a thing called The Conversation. And Esme's boss wrote an article in The Conversation about phages and antibiotic resistance. And so I wrote to him and said, well, would you like to come and give us a talk to Café Scientifique? His reply was, Esme will do it. So Esme is here. So Esme is, well, she'll tell you what she's been doing and a bit of her background, but she's running quite a really nice sort of citizen science program in Southampton about getting around antibiotic resistance, and that's something we should all be concerned about. So, Esme, the floor is yours.
Thank you. Good evening, everyone. Can everyone hear me okay? All right. Thank you for the introduction. My name is Esme, and I am hopefully the project coordinator for the Phage Collection Project, which is a citizen science initiative based at the University of Southampton. And today I'll be discussing a little bit about our research and how we're hoping to use these viruses, known as phages, to overcome and tackle antibiotic-resistant infections. Before I delve into the talk, though, I thought I might get a little bit of public participation and ask a few members of the audience what you think the similarity might be between a toilet and a hospital. Any ideas? Yeah, a lot of bacteria. That's a common answer. Yeah. Now, what if I actually said to you that your toilet might be able to save your life in the future if you contract a bacterial infection? I know it might sound a little unorthodox and a little bit strange, but I'll explain later on in this presentation. I think when we normally think about treating a bacterial infection, the first thing that we'll think about are antibiotics. Now, antibiotics are a particular class of drug that have been designed to kill bacteria or prevent them from replicating. Now, whilst we've used antimicrobial compounds for centuries now, I think we typically associate the discovery of antibiotics with Alexander Fleming, who discovered penicillin in 1928. Now, it's been argued by many that the discovery of antibiotics was the greatest medical breakthrough of the 20th century. Before we had antibiotics, if you even had a small cut or incision, like a paper cut, if that became colonized and infected with a bacteria, it could potentially kill you. Not only that, antibiotics have made routine medical procedures, such as cesarean sections or going to the dentist for a filling, much more safe to complete. So they really have helped to revolutionize our healthcare. And there are many different ways and many modes of actions that antibiotics can use to effectively kill our bacteria causing an infection. And I thought I would just briefly explain 3 mechanisms to you this evening. So the first mechanism that some antibiotics, like tetracyclines, methicillin is one you might have heard of, use, is that they stop protein production. So when the antibiotic enters the cell, it binds to an organelle in the cell known as a ribosome. And the ribosome is responsible for creating and synthesizing essential proteins which are needed for the bacteria's survival and replication. By binding to the ribosome, it stops the production of these essential proteins, which causes the cell to die and helps us again to eliminate an infection. A second mode of action that antibiotics like penicillin use is that they stop the synthesis of the cell wall. So in the case of penicillin, what penicillin does is it prevents the synthesis of a molecule called peptidoglycan. Now, peptidoglycan is a complex polymer molecule made from sugars and amino acids, and it's an essential molecule used in the bacteria's cell wall. Now, when you stop and inhibit the production of peptidoglycan, what you do is you create a more unstable cell, and the cell becomes more weak, and it's more likely again to lyse and die, again, which is great because it enables us to successfully eliminate the infection. And a third method that some antibiotics use is that they prevent DNA replication. So a fluoroquinolone, such as ciprofloxacin, what it does is it binds to these molecules known as enzymes, which are essential for the bacteria DNA to replicate, which helps it to produce offspring and proliferate. By preventing this DNA replication, again, you prevent the bacterial cell from reproducing, and again, it eventually dies, and we again get this bacterial clearance. So these are, some of the common modes that antibiotics will use. Now, unfortunately, and this is why I'm here this evening, is we're facing a major problem, and it's now that antibiotics are becoming much less effective at killing our bacteria. And this is what we call antibiotic resistance. So antibiotic resistance is when bacteria develop mechanisms and ways to overcome the actions of antibiotics, enabling them to survive and proliferate and cause chronic long-term infections. You may have also heard of this term as antimicrobial resistance, but this term tends to be more broad, encompassing different types of microorganisms such as viruses and fungi. Now, for us in the lab, it's actually quite easy for us to demonstrate antibiotic resistance. So here we have two agar plates, and the white discs that you can see are paper which have been soaked in different types of antibiotics. And if you just look at the plate on the right-hand side, you can see this kind of cloudy looking layer on the agar plate, and this is our bacteria. It's our bacterial lawn that's been growing. So if you go back to the plate on the left-hand side, we can see that we don't have any bacteria growing. This is because the antibiotics have successfully killed the bacteria, which is great. When we see this in the lab, it means we have a bacteria that is sensitive to the antibiotic. And if there is a patient suffering with an infection caused by this bacterial strain, we can successfully kill it with one of these antibiotics. However, if we then take a look at the plate on the right-hand side, we can still see that we've got this bacterial growth. A few of the antibiotics, we can see this clear zone, which is the zone of inhibition. So they have been able to kill some of the bacteria, but not as effectively. So when we see an agar plate like this is quite a concern for us. It demonstrates to us that this bacteria is resistant to antibiotics. And if we have a patient who is suffering from an infection caused by this bacterial strain, we need to start considering potential alternative therapies that we can use to successfully treat and clear their infection. Now, I think in many cases with antibiotic resistance, a common misconception is that it's us as humans that develop resistance to the antibiotic. And actually what it is the bacterial cell itself developing the resistance. And again, there are many, many different ways that bacteria can do this, but I thought I would briefly summarize 3 for you this evening. So the first mechanism that bacteria can use is that they use these protein transporters known as efflux pumps, which sit within their cell surface membrane. What they can do is they can upregulate these, and they can force the antibiotic out of the efflux pump before it's carried out its mode of action, meaning this, the antibiotic doesn't work and the cell can go on to survive and proliferate. The second mode of action that bacteria can do is they can actually just prevent the antibiotic even entering the cell in the first place. They do this by down-regulating molecules known as porins, which again sit within the cell membrane of the bacteria and normally are what the antibiotics use to enter the cell. By down-regulating these molecules, the antibiotic cannot enter, which again prevents it carrying out its mode of action, enabling the cell to survive. And then a final mode of action that bacteria can use is that they can actually modify and inhibit the drug once it enters the cell. So you might have heard of these molecules called beta-lactamases. And these, again, are enzymes within the cell. And what they're able to do is they're able to cleave the antibiotic molecule. And by cleaving it, again, it renders it ineffective. It no longer works, and it can't carry out its mode of action. So these are some, when we sequence bacteria, these are some of the mechanisms that we can observe that they are using to develop resistance to these antibiotics. And again, this is a major concern for us when we see this in the lab. And the problem of antibiotic resistance, it's been around for decades, but it really is becoming a global health threat. So it's been estimated that by 2050, 10 million people each year could lose their lives due to antimicrobial resistance, which is a significant number. We also need to consider the economic consequences of it too. So currently it's estimated that antimicrobial resistance costs the NHS £180 million per year. And actually in the next 25 years, global economic losses are expected to exceed 100 trillion US dollars. So it truly kind of highlights the problem of this threat. And we need to expect that these numbers are going to continue to rise if we don't find alternative methods to treat these infections. And I think truly the, problem was really realized back in 2019 when the World Health Organization actually declared antimicrobial resistance as one of the top 10 global health threats facing us as a society today. So it's something we really need to think about and be aware about, and as I said, try and find solutions to tackle this problem. I think another as well big misconception with antimicrobial resistance is that it doesn't affect maybe us here today in this room, but unfortunately it does. And locally to us in Southampton, we're really seeing the problem of antimicrobial resistance. So I thought I would provide this graph, and this is a graph showing the number of patients suffering with a bacterium known as non-tuberculosis mycobacterium. Non-tuberculosis mycobacterium can also be referred to as NTM. And NTM typically causes lung infections, and unfortunately, it's normally the elderly that are most at risk from this bacterial pathogen. So along the x-axis, we've got our different species of NTM, and then on the y-axis, you can see the number of patient cases. And you can see that just locally to us from Southampton General, that we have hundreds of patients struggling with these bacterial infections. And unfortunately, in many of these cases, these species demonstrate resistance to antibiotics. The patients therefore have chronic and recurrent infections which need to be treated. So yes, unfortunately, it is a problem that could affect all of us in this room today. And again, it's something that we really need to address. So we clearly need to find a solution. We need to think about alternatives. And this is where our phages come in. So phages are viruses which infect and kill bacteria. Unlike other viruses that we typically hear about, such as COVID, for example, phages do not infect and kill our human cells. They do not recognize them, so they cannot kill them. I've been told a lot that these phages look a lot like lunar lander rockets. A lot of people have that thought when they've seen them. But here we've got a transmission electron microscope of a phage, which infects and kills Klebsiella pneumoniae, which is a species of bacteria which typically demonstrates resistance to antibiotics. Now, phages are extremely abundant. They're actually the most abundant biological entity on this planet. They can be found in pretty much any environment. So they can be found in soils, they can be found in rivers, in lakes, and they can even be found in the human gut. They're on our skin right now, so they're constantly around us. For something that's very small and typically unknown, they are constantly in our lives. And whilst they come in a huge variety of shapes and sizes, when we normally isolate phages in our lab, we observe these three phage morphologies. And these morphologies sit under a class of phages known as the cordoviruseps. So we have our myophage, our cyphophage, and our podophage. And whilst they may look slightly different, they share structural similarities. So first we have this grey structure on the top, and this is the capsid head of the phage. And what the capsid does is it contains and protects the phage's genetic material from the surrounding environment. In green here, you can then see we've got this tail-like structure, and at the end of the tail, we have these tail fibers. Now, the tail fibers are almost like the legs of the phage, and the phage uses the tail fibers to recognize and bind to complementary receptors on the bacterial cell membrane. And once it's found these receptors, it can then bind securely and infect that bacteria. And as I said, phages are extremely abundant and diverse, and actually we can just observe this in the lab even from one sample. So here, this is a transmission electron microscope of a virome which we collected from a wastewater treatment plant. And if you zoom in to this one sample, you can see we have a variety of different phages and phage morphologies. And actually this one in the top here is what we call a megaphage, and hopefully you can see it on the screen, but it's much bigger than all the other phages. So we really get a huge diversity, and this is just in one sample from Portswood, I think. And I thought I would just include these facts because phages aren't typically well known, but actually, as I said, they play a huge part in our daily lives. And I'm not sure who quantified these, but phages are actually the greatest predators on Earth. So they kill around 40% of bacteria that reside in our oceans every single day. And again, who found this? I don't know, but there's more phages on Earth than stars in our galaxy. So it just highlights how we encounter them every single day in our lives. We just might not see them or know about them. So how do our phages actually infect and kill the bacteria, and how can we actually apply them therapeutically? So as I mentioned previously, our phage has tail fibers, which are like the legs of the phage, and it uses these tail fibers to recognize complementary receptors on the bacterial cell membrane. When it recognizes these bacterial receptors, it binds to them irreversibly and docks securely, and then it releases the genetic material illustrated in red here into the bacterial cell membrane. Once the genetic material has entered the bacterial cell, it hijacks the bacteria's host cell machinery, and it causes the bacteria to replicate and produce new components of the phage, so its capsid, its tail, and its genetic material. Once all of the different structural elements of the phage have been formed, they then come together to assemble new virions within the bacterial cell membrane, within the bacterial cell. What they then do is produce two other molecules called holins and endolysins. And what these two molecules do is they break down and degrade the bacterial cell membrane, releasing the new viruses into the surrounding environment, killing the bacterial cell, and enabling the new viruses to go into the surrounding environment and infect and kill more bacteria. And we can actually, again, see this nice and clearly in the lab. So here we have some cells, cell green, and these are E. coli cells from a clinical isolate that we received. And we can see cells here in green demonstrate that they're living cells. When they turn red, which you'll see in a minute, this indicates that the cell is dying. So when we add our phage sample, what you can see is we can start seeing these cells turn red, and actually we get a reduce and a decline in the bacterial population, indicating to us that we've successfully found a phage which is able to infect and kill the bacteria, and in this case could potentially be used to treat this clinical strain. So how do we find these phages, and how do we as a lab try and find phages that can successfully treat and save a patient's life? So first we have to find our phages. Now, as I said, phages are extremely diverse and they can be found in very many different environments, soils, rivers, oceans, but we typically isolate ours from wastewater. It's not a very glamorous job. Normally a PhD researcher has to go with a big plastic tub and collect 20 litres and bring it back to the lab. So it's not a glamorous thing to do. But the reason that we use wastewater is because wastewater is more likely to contain pathogenic bacteria, and therefore we're more likely to find phages in these environments which are able to successfully infect and kill these bacteria. And this is how your toilet could potentially save your life in the future. You never know, what could be in this wastewater and potentially save you. Once we bring our sample back to the lab, the first thing we do is we call something called a spot test. And this is just to determine whether this water sample that we've collected does actually contain phages which can infect and kill clinical isolates. Now, to us in Southampton, we have a particular focus on the antibiotic resistance strains that typically cause infections for patients, and you might have heard of these as escape pathogens. So these are six species of bacteria that typically display resistance to antibiotics. So what we do is we first grow our bacteria on an agar plate, and again you can see this kind of cloudy looking layer on the plate. This is bacteria, and in this case it's again Klebsiella pneumoniae, which has come from a patient. And then what we'll do is we just spot samples of our water sample onto the plate, and then we leave it for a few hours. And when we start to see these hopefully you can see these clear markings on the plate, this indicates to us that a phage is present because it's been able to eat and clear the bacteria that have been growing. So when we see this, it's a really good sign. It means we have a sample that does contain phages and that we can hopefully then purify these phages and use them to treat this patient. So I think this is a phage called ROF90, which has been able to successfully, ROF47, which is able to successfully kill this bacterial strain. So once we've identified this phage, we then actually want to determine how good it is at infecting and killing the bacteria we're interested in. So here I've presented two graphs, and these are demonstrating growth curves. So for simplicity, when the line goes up, it indicates we've got a happy bacteria. The population is growing over time. It's nice and happy and healthy. But then if we add our phage, what you can see is we get this initial growth and then plateau. And when we see this, this indicates that, again, we found a phage which is successfully able to infect and kill and reduce the bacterial species. Now, as I said, phages are extremely specific. So normally phages only infect and kill a certain repertoire of strains within a bacterial species. So we might have found a phage which infects and kills this orange bacteria. But when we test it against a different strain, here highlighted in green, you can see even when we add our phage, the bacterial population still increases. So this indicates to us that this phage would successfully treat a patient that is suffering from this first bacterial strain, but not the second. And this enables us to accurately work out the host range of our phage. Once we've conducted this for a whole biobank of phages and clinical strains, we then plate this as a heat map. So across the top, we have our different clinical strains, and then along the side, we have our different phages. Now, the squares in orange indicate that the phage has been able to successfully infect and kill the bacteria. In white, this is where we've not seen the plaque on the agar plate, so this indicates that we've not, this phage isn't able to kill the bacteria. And then we have this lysis from without, or actually what I should now refer to it as undetermined lysis. And this is where we've seen a bacterial, we've seen these plaques on the agar plates, but they normally don't look as transparent, they're slightly opaque. And this indicates that the bacteria has died, but it's probably not due to the phage infection, and it's another mechanism that's caused this. And by producing this heat map, we can then quickly determine which phages are great therapeutic candidates. So, for example, if we had a patient suffering with this strain here, 56, we can see a few phages, there's 6 phages here that can successfully infect and kill it, and then we can identify that and hopefully give it to a patient. And what I thought I'd do is actually provide an example of a patient case that we actually received as a lab. So a PhD student in the lab received a strain of Pseudomonas aeruginosa. Now, this patient had a prosthetic aortic graft surgery, and unfortunately during the surgery contracted an infection caused by the strain of Pseudomonas. So the initial test that we did, as I mentioned, was this test called a spot test. So we had 2 wastewater samples, and one of them is here and one of them is here. And when we plated them out, you can see that we got a few plaques in the first sample, but we didn't in the second sample. So again, this just highlights phage diversity and the importance of collecting lots of different samples, because unfortunately in a lot of cases, some of the samples we collect actually aren't suitable for the strain that we're interested in at the time. And these plaques here that you can see on the top, again, when we saw them, it is a good sign. It means we have a sample that can infect and kill this bacteria, but they're not very clear. Again, they're not that big. So when we see this, we think the phage maybe isn't that effective at infecting and killing that bacterial strain. So what the PhD researcher did was she then trained the phage. So what you can do is you can train the phage to make it better at infecting and killing the bacteria. So what she did was she just kept exposing it to the bacteria, and over a 3-day period, you can see how the phages became much better at infecting and killing this strain of interest. So much so that you can, again, she diluted it and you can see these individual phage plaques. So again, we were able to find some phages for this patient's infection, and increasingly more and more we've seen, even kind of as I've been in this role for the last year, we're having more kind of interest in patient cases, unfortunately, who are struggling with chronic bacterial infections. And yeah, there's definitely more of an interest to receive phage therapy as a treatment. Now, phage therapy isn't a new thing at all. It's not something that we've come up with as a lab and can take credit for. It's been around for a really long time. So phages were actually thought to first be discovered in 1896 by a bacteriologist called Ernst Hankin, who was observing cholera bacteria in the river Ganges. And he noticed there was something present in the water that was able to kill these bacteria. It wasn't until 2 decades later where phages were independently described by Frederick Tort and Felix de Hull. And actually, Felix de Hull was the first person to apply phages therapeutically in 1919 to treat patients suffering from dysentery. So it's been around for a really, really long time now. Now, the reason that phage therapy isn't so commonly known is when antibiotics were first discovered, especially within the Western world. That was kind of what drove interest, and we kind of forgot about phage therapy. In Russia, Georgia, Poland, they actually continued to experiment with phage therapy and use it to treat burn wounds and patients. And so much so that in Georgia, for example, phage therapy is a routine treatment, and you can actually get it over the counter. But with the presence of antibiotic resistance and the emergence of this threat, we're now seeing a real interest from the Western world, and kind of as a whole global society, there's much more interest in this field. Now, there are many different benefits to phage therapy, but I thought I would go over a few this evening, just a couple. Now, the first is that phages, as I said, can kill antibiotic-resistant bacteria, and that is because our phages use different modes of action to our antibiotics. So when our bacteria develops a mechanism to become resistant to an antibiotic, it can still be susceptible to our phage. So this is why we can administer it to patients struggling with these chronic infections. A second benefit, as I said, of phages are extremely specific. They only infect and kill a few strains within a species of bacteria. Now, this is really great because then when it means we come to designing a therapy for a patient, what we can do is we can select phages that only infect and kill the bacteria responsible for causing the infection. So unlike antibiotics, which are broad spectrum, killing both the good and the bad bacteria, they can cause things like gut dysbiosis, and this can unfortunately cause side effects like diarrhea when you're on this medication. But when we tailor the phages, we can, as I said, tailor them to only infect and kill the bad pathogens, leaving our, for example, gut microbiome intact, which is a real positive. The third is that phages can break down biofilms. So biofilms are basically complex microbial communities of microorganisms which are embedded almost within this glue-like matrix known as the EPS. And what the EPS does, it helps to protect and harbour the bacteria from antibiotics. The EPS stops the antibiotic reaching the bacterial cell, again enabling them to proliferate and continue to survive. And in many cases, biofilm infections, again, I think are between up to 1,000 times more resistant to antibiotics compared to free-living bacteria. So they can cause really stubborn infections for patients, particularly cystic fibrosis patients when they contract a lung infection. Phages are able to produce an enzyme which is able to break down and degrade the EPS, which then helps the antibiotic to reach the bacteria and then successfully treat that infection. And another benefit is that phage therapy can actually be administered as a combination treatment alongside other therapies. So whilst I'm here talking about phage therapy today, I also think it's quite important to highlight that whilst antibiotics, unfortunately, we are seeing a decrease in their efficacy, they're still extremely important in healthcare, and they're something that we're going to rely on still for a very long period of time. And actually, in many cases, it's the best course of action for a treatment is to receive a combination of phage therapy and the antibiotic together. And again, we can demonstrate that quite clearly and nicely in the lab. So here we have, again, an agar plate, which has been streaked with a species colony of bacteria. And this is just our control plate, so when the bacteria has not been treated. Now, when we treat it with the antibiotic Rifax, we can see a decrease again in bacterial growth, suggesting to us that the antibiotic has been able to kill some of the bacteria. And again, when we use phages 2 and 6 to treat the bacteria, again, we can see a decline in the bacterial population. But as you can see, we still do have this growth here. So it indicates that they can kill them, but not completely eliminate. But when we actually administer our phage and antibiotic together, you can see we almost have complete clearance of the bacteria. So actually, again, in many cases, it's best for us to administer this as a combination therapy. Now, there are obviously considerations with any therapy, and there are some that we need to think about when we're designing and administering phage therapy for a patient. And the first is immune recognition. So phages, whilst they do exist naturally in our gut anyway, on our skin, when we administer phages to a patient, there is a chance that the immune system can recognize them and attack them. And this creates two considerations. The first is when we administer our phage, the immune cells can recognize it and clear it from the body before it's even reached the bacteria that it is causing the infection. This means that the phages don't even have an opportunity to infect and kill the bacteria, so the infection continues. And obviously, with any immune reaction, there is the chance that there could be side effects for the patient and adverse effects. Now, in the literature, in compassionate use cases of phage therapy, strong immune reactions haven't been observed. But in order to truly kind of assess this, we need to have robust clinical trials with large cohorts of patients to truly understand this consideration. And something else that we also have to consider is phage resistance. So like bacteria can develop resistance to antibiotics, they can also unfortunately develop resistance to phages. And we can refer to this as an arms race, because bacteria will develop resistance mechanisms against phage, but then fortunately, phages can also evolve and overcome the resistance mechanisms of the bacteria. So it's kind of, this constant battle that's been going on for millions of years. But the bacteria, what they'll do is they can develop defense systems against the phage, which can stop it either infecting the bacteria in the first place, or it can cleave and the phage's DNA when it enters the cell. But then phages can evolve and overcome those mechanisms of those bacteria to survive and continue to proliferate and kill the bacteria. And another positive thing, obviously, with this is that we typically observe in many cases that when a bacteria develops resistance to a phage, it normally then becomes susceptible to antibiotic treatment. And the reason for this is because it takes a lot of energy for the cell to be resistant to both antibiotics and phages at the same time. So this is when, again, administering the therapy as a combination with antibiotics is great, because it's likely that you might have an infection with bacteria which are resistant to both antibiotics and phages. So kind of giving it this double hit means you can successfully try and eliminate all those bacteria causing that infection. Now, as I said, phage therapy was kind of forgotten about when it was discovered over 100 years ago, especially in the Western world. But with antibiotic resistance becoming a more pressing threat, we have seen a real interest for phage therapy. And as I said, even just within my role in the lab in the last year, I've seen a real momentum increase in policy, but also amongst the general public for this therapy. So currently in the UK, phage therapy is only administered on a compassionate use basis. So this is when all other treatments have failed for a patient, or they're not suitable maybe for that patient at the time. And they will then, their infectious disease specialist can then look into administering and applying phage therapy to that patient. Now, the government, as I said, with antibiotic resistance is really recognising the potential of phage therapy. So there was actually a report that was published by the House of Commons Science, Innovation and Technology Committee in 2024, which highlighted the potential that phages have to help us tackle this threat. And it was also actually recognised in the UK government's latest national action plan to address AMR, with a push to see more investment and research into this therapy. And we're seeing more clinical trials actually popping up for phage therapy too. So Horizon Europe back in May released a call for a phage therapy clinical trial, and that will take place across Europe. And we're seeing a lot more now happening in the United States too, and in different countries around Europe. So it really is becoming a much bigger thing, and these clinical trials will help to see phage therapy be included within routine healthcare procedures. And the MHRA, who are the Medicines, Healthcare and Regulatory Agency, also published a document last year, which is to help phage researchers and pharmaceutical companies interested in developing phage therapy learn about the regulatory guidance and mechanisms that they need to follow in order to successfully bring these therapies to market and make them more accessible for patients. Now, I guess I thought, again, I get a bit of public participation from the audience. So some of you may have heard about phages and phage therapy before attending this talk, but I thought after hearing some of the things I've spoken about this evening, just with a show of hands, who would be willing to receive phage therapy if they contracted a bacterial infection? It's positive to see. And who would be maybe less willing or a bit apprehensive? Fair. One, I think that's okay. We can have a conversation at the end. Maybe I can see if I can convince you. Now, as I said, this is research that we're actively doing at Southampton, that the PhDs are looking into, but we've also developed a citizen science project called the Phage Collection Project. And this is to increase awareness about phage therapy amongst the general population, but to also hopefully engage and empower members of the public to contribute and get involved in our research. So the Phage Collection Project has many different elements to it. One of those is our willingness survey. So we're really interested to learn about members of the public's views on phage therapy, antibiotic resistance, and potentially things that would impact their willingness to receive and accept this form of therapy. We've also developed a phage sampling initiative where members of the public can collect a 50mm water sample from their local environment. So this could be a river, a pond, a lake. We very much don't ask that you collect a sample of wastewater. Please keep it to environmental samples. And then you can send them back to our lab and we can try and isolate a phage from them. And if we're able to find a phage which infects and kills one of the bacterial species we're interested in, we can let you know and you can name that phage too if you're interested. And we also work locally with a few schools. So we work with a few colleges, and we've designed a phage isolation course. So college students 16 to 18 who are studying A-level science can, again, try and find phages and actually isolate them in their school labs. So thank you so much. I think I might be ahead of time, but thank you so much for listening. If you're interested in learning more about the project, you can scan the QR code or speak to me later on. And thank you so much. I hope you've enjoyed the talk.